Okay, folks, let's move on to the next presentation. It's a pleasure to welcome Karen Shapiro. Karen's actually new to Ontario. She recently moved to Guelph with the family from the University of California in Davis, and she's moving into the University of Guelph along with her husband at the moment. But she's going to talk to us about the work that she carried out on Toxoplasma in sea otters in California. So Karen, welcome.
Thanks so much, Andrew, for the introduction. So I'm delighted to be here and share just a little bit, a glimpse of some of my work that I've been doing for the past 10 plus years at UC Davis. So when I started, when I joined the research team at Davis, it was about 2004. They were focusing on diseases in sea otters. And I just wanted to start with a couple of natural history facts about sea otters to help orient you for the rest of the talk. So sea otters are exclusively coastal, meaning they live within 500 meters of the coast their entire lives. So very unique from some other marine mammals. Their ecology is also very close to that of kelp forests. They're one of these classic keystone species. You remove them from the system, you'll see a kelp forest basically become what they call urchin barrens. So it's like a desert under the sea. So you see complete ecosystem changes if you take them out of the system. And another important fact that's quite interesting is that the pups learn what prey to eat from their moms, and they become specialized on specific invertebrate types. And they have to eat a lot. They don't have this blubber layer that other marine mammals have to keep warm, so they have to eat about 1/3 of their body weight each day just to keep warm in the Pacific coast. So sea otters are currently classified as threatened after they're almost completely decimated due to the fur trade in the 1800s. And even though their northern cousin up in Washington and Alaska has been doing really well, the southern sea otters have not been able to recover as well, and they haven't been able to reach over this 3,000 level, which would allow their delisting. So ecologists started to look a little bit more about what was causing this kind of stagnant population growth, and they found out that prime age young adults' deaths due to infectious diseases was one of the primary causes. They looked further and they found that one specific pathogen, Toxoplasma gondii, was killing almost 1/3 of sea otters. This was a study that was done between 1998 and 2001. So the puzzle here is that Toxoplasma gondii, as far as we know, is a terrestrial pathogen, meaning that only felids, only cats, both wild and domestic, can serve as a definitive host where the parasite can complete its life cycle and shed oocysts in its feces. There's a lot of intermediate hosts. Almost all warm-blooded animals that we know can serve as intermediate hosts. They're vastly successful in, globally, and potentially one of the most successful human parasites. It's thought that about 1/3 of the entire global population is infected. Now, seroprevalences really vary depending on where you are, as well as in socioeconomic status. So just a couple of examples to show you the range. In the United States general population, so just a cross-section, you have about 15,000% of people infected. But just north in Canada, some populations, native populations, more than half of people are infected. So about 60% seroprevalence rates, way higher. There are three main transmission routes. The first one is kind of the classic transmission route, and that would be the foodborne route. So a typical way by which a cat gets infected is when it eats a definitive host, like a rodent or a bird that has these tissues in its brain and muscle. Also a classic way by which people can get infected from undercooked meat. Congenital infection is another important route of infection. So this happens classically when a woman becomes infected for the first time. She can pass the parasite transplacentally to the fetus. And then finally, what is going to be the focus of this talk is oocyst ingestion as a route. So cats become infected, they shed these hardy oocysts in their feces, and this is the stage that we think infects sea otters, and it's also an important route of infection to people who can become ill from consuming seafood that's contaminated or through contaminated water. Okay, so let's get back to the puzzle. We have this terrestrial host, we have these marine mammals that are infected, and we have these oocysts that are somehow moving from land to sea. But even though we can imagine that cat waste can end up in the ocean, it's a huge ocean out there, right? So how can so many otters become infected and die from this parasite? The first clue that we obtained was actually from some of my PhD studies when I was looking at the surface characteristics of oocysts that determine how the parasite moves in water. One very important characteristic is surface charge. And what we found out is that if you put oocysts in fresh water, they're negatively charged, meaning that they're going to repel other similarly charged particles, and they're likely existing in seawater as suspended particles. But if you put oocysts in either estuarine or marine waters, their charge is neutralized, meaning that they're more likely to clump and aggregate. So something very interesting happens to their surface charge as you move from fresh water to marine waters. Why is this important? Why is aggregation even important and interesting? It's very interesting because an oocyst that is singly floating in water is a completely different type of particle than an oocyst. I know it's hard to see, but here's an oocyst here that's embedded inside this marine aggregate. Oocysts that are just existing in the water column are likely to go where the water goes. They're very light and buoyant. An oocyst that is in an aggregate is going to sink. If it sinks, it's going to accumulate where this aggregation process occurs. So if we understand how Toxoplasma aggregates, we can better predict where these oocysts are concentrating in the environment, and therefore we can better predict where hosts can become infected, where the risk is greatest. It also gives us clues on bioavailability. So a singly floating oocyst like this one is not likely to be ingested by anything that otters eat or that we eat. But a lot of organisms out there eat marine snow. This is a source of carbon, a source of energy. So if we know where and when these oocysts become entrained in aggregates, we can also tell when seafood, when certain marine invertebrates get contaminated. And then finally, most models out there that estimate where waterborne infectious agents go assume, they track water molecules. They assume that pathogens go where water goes, and that could be completely inaccurate if you take into consideration the fact that pathogens can interact with other particles and sink in marine aggregates. So to ask some specific questions about aggregation, we became very interested in a group of molecules that's called TEP. TEP stands for transparent exopolymer particles. Kind of a mouthful. It's basically biofilms. So it's pieces of biofilms that are suspended in the water column, and they can be, they're very gelatinous, they're very sticky, and they basically hold marine snow or marine aggregates together. Sorry, I forgot to define snow here. Marine snow and marine aggregates are synonymous. And these TEP molecules are produced by phytoplankton, cyanobacteria, and kelp, which is why we're so interested in it. It's exactly where sea otters live. So the specific hypothesis we wanted to test is that the association of Toxoplasma with marine aggregates will increase as a function of this gelatinous sticky molecules called TEP. So the first experiment we conducted was we took a seawater sample and spiked it with increasing concentrations of alginic acid. Alginic acid is one type of a TEP that oozes here from giant kelp, which is a type of kelp that we have in California. So if you guys ever touched kelp, you know it's really slimy. That's basically TEP. The experimental results were really straightforward. We have here on the y-axis the proportion of oocysts that became entrained in aggregates versus the concentration of TEP. Very strong linear relationship. So yes, the data does support the hypothesis that with increasing TEP, you have increasing association of Toxoplasma with marine aggregates. Okay, so that's very interesting. The pieces of the puzzle are starting to fall into place. These experiments show that yes, you have more marine snow, more marine aggregates settling where you have a lot of TEP, which happens in kelp forests, which is where sea otters live. But that couldn't explain the full story because many kelp-dwelling invertebrates eat snow. But not all sea otters are infected. In fact, the only dietary source that's been identified as a risk of infection is turbine snails. So those sea otters that specialize in eating snails are 12 times more likely to be infected with Toxoplasma. And the thing that sets turbine snails apart is that they graze on kelp directly. They can eat marine snow, marine aggregates if it falls and becomes attached to kelp, but a single floating aggregate is not accessible to a marine snail. So the second set of experiments was done to see if oocysts can actually adhere to kelp directly. So we did a series of experiments with different tanks that are not showing up. So it's not moving forward. I don't know why. So I'll start talking so I don't run out of time. So we had 4 different treatments. Can you hear me here? Okay, I'm kind of yelling. So we had a treatment with seawater and real kelp. So we have TEP in the seawater and TEP attached to the kelp surfaces. So there's TEP that exists from phytoplankton in the seawater, but also directly on the kelp surfaces. We had treatment B, which is filtered seawater and real kelp. No TEP in the water, but TEP on the kelp. You guys kind of following me? We had treatment C, which has filtered seawater and model kelp, plastic kelp, so no TEP on the kelp, no TEP in the seawater. And a treatment D, which just accounted for oocysts being lost in the container that had no kelp and just filtered seawater. Should I just continue?
Just keep going.
And so what the results showed is that both in treatment A and B, 1/3 of the oocysts that were introduced into the tanks became associated to the kelp surfaces, meaning that all you needed, here's treatment A, B, C, and D, thank you so much. Oh wait, don't go, it's not going anywhere. So in treatment A and B, at least now you can see the treatments, 30% of the oocysts became attached. So this is telling us that association can occur in substantial numbers due to the TEP on the surfaces of kelp.
I'm so sorry. I'm so sorry.
So nothing to do.
I'm sorry.
Do you want to try restarting it?
We might run out of time is the only thing.
Okay.
Okay.
So next set of experiments. Do you want to restart the program, or do you want me to?
Yeah, maybe. Yeah, just restart the program. It's probably the safest bet.
Next, so now we know that oocysts can accumulate in marine snow where sea otters live. We know that the oocysts can attach to the kelp, but what about the final link, which is the snails, right? So we wanted to see if snails can actually pass the infection to sea otters, because nobody has looked before to see if marine snails can be a transport horse for zoonotic pathogens. So we did an experiment where we exposed a cohort of snails to Toxoplasma in one main tank, and then after a 24-hour exposure period, we divided the snails into what we called snail condos. So each snail was individually monitored in a plastic container with kelp and filtered seawater for 2 weeks. And the results showed that Toxoplasma was being shed in the snail's feces for 11 days after we removed them from the contaminated Toxoplasma source. We also were able to show that the concentration of oocysts in the feces was 2 to 3 orders of magnitude greater than the concentration of Toxoplasma in the seawater. So we're able to verify that Toxoplasma was that snails were an efficient paratenic host for Toxoplasma because they increase the duration of risk for otters, and they increase the magnitude, the concentration of oocysts that any particular otters can be exposed to through bioconcentration. So that was really the end of the data that I was going to show, and then I had a couple of really neat summary slides, basically driving the point that we're able to now put the pieces of the puzzle together, showing that the reason why we think so many sea otters are infected is because you have these 2 distinct but not mutually exclusive pathways for these oocysts to become concentrated where sea otters live, right? You have the marine aggregation that's happening, and you have this association with kelp. And if you have more kelp, you're going to have more marine snow because you have more TEP. If you have more oocysts in marine snow, you have more of them settling on the kelp, becoming accessible to snails. So the snails become exposed through the association of Toxoplasma and kelp, and provide this parasite to the sea otters. And then I also propose that the similar mechanisms might be at play for how other marine mammals, for example seals here in Canada, could become infected and serve as a potential foodborne risk to native communities. And then I have one more slide just driving home the point that it's not just sea otters and marine mammals that are potentially infected, but also people. So people can become infected through contaminated seafood and potentially through recreational contact as well. And that was the end of my presentation. I'm so sorry for that malfunction.
don't know if I'm at the end there. Yeah, that's kind of the slide. It's the slide with the video.
No, the video
No?
is from here.
Oh. It's going to do it again then? Yep, same thing.
Okay, folks, let's join Karen. Thank Karen. Does anyone have any questions? Karen, sorry about that.
Cheapers, yeah.
Anyone have any questions? Karen, I had one question, and that is, do you believe the problem is a relatively new phenomenon? Like historically, why has it become a problem apparently recently, or is it only an apparent problem recently?
Yeah, that's a great question. I think twofold. I think that we're finding it because we're looking for it. So it's this classic phenomenon in epidemiology with surveillance, right? If we are not looking for it, we're not going to find it. But I do think that there's things that we have done anthropogenically to increase the likelihood of Toxoplasma reaching the coast. So we actually have an entire program that's looking at the impacts of climate variability and habitat change and pathogen pollution in California. There's a set of experiments that I've done, again, as part of my PhD work to show that wetland degradation increases oocyst Toxoplasma transport to the ocean by orders of magnitude. And in California, we've lost about 90% of the wetlands due to urban development. So we also have the increase in domestic cat populations, and we have untreated storm drain runoff reaching the oceans. So I think it's probably both. I think it may have existed in the past, and we didn't know about it, but I do think that the conditions that we have created in terms of altering our coastal ecosystems are such that they're enhancing the likelihood of waste like cat litter or cat feces and whatever pathogens are in there reaching the coast.
Okay. Any other questions? David?
Based on your work, what types of seals do you think we should be looking at in the north? Should we be looking at walruses that mostly feed on mollusks, or seal? Where would you go?
So the question was which marine mammal I would focus on in terms of looking for infected marine mammals here in the north. So bear in mind that what we found in sea otters is that actually mollusks weren't the main risk food. We thought they were initially because we're able to document in the lab that oysters and mussels can concentrate toxo, and they can, but that wasn't the food that was providing the most risk, at least not to otters. So I think each system is probably going to have its own unique food web dynamics that may enhance or reduce the risk for certain marine mammals. So I wouldn't necessarily focus on specific, on marine mammals that hold a specific niche in terms of a diet. I think I would first ask where the problem is, who is getting infected, and then look to see what about that life history of the animal makes it most at risk. Answer your question.
Any other questions? Doug?
Since the problem seems to stem from cats, is there anything that can be done as an intervention on the cat side of things?
That is a hugely politically charged question. We've tried, and we are trying, and it's very difficult because it's a very emotional topic to suggest certain management practices, which is really what you should do for feral cats or outdoor cats. So we've had discussions with communities and with SPCAs. I can tell you what I think should be done, but which is remove all outdoor cats, if as much as possible. The likelihood of that happening, I think, are very, very low.
Any other questions? If not, can you please join me in thanking for that presentation? Thanks. Sorry.